so as promised, I'm going to go through this pretty nice and quickly, and hopefully it won't be too bad. My best recommendation is to go back and look over your notes and any homework we did, and the textbook reading, the copy that I made for you guys that you guys read. That should help a lot, but this is also a fantastic way to review for the test, so let's go through it. Alright, the first one, results in diploid cells. What you need to remember, guys, diploid means that you have homologous chromosomes. So you've got two copies, one from mom and one from dad. So even though the chromosomes aren't identical, they have the same types of genes. They're just slightly different. So they have alleles on them. So for diploid cells, that's what almost all your cells are. And the way that you grow, the way that your cells duplicate is by a process called mitosis. And so the whole point of mitosis is you have one original cell, you create two identical cells. So you're replicating that same cell but making two exact copies. So you still have diploid cells. Now, for the second one, results in haploid cells. That's the whole point of meiosis, is to create cells that only have one copy. So they're split, so they're haploid. The only cells where you have meiosis in your body are your reproductive cells. So for men, sperm, and for females, they have eggs. Sorry, my brain. All right, occurs in humans. Well, both of these processes occur in humans. Mitosis, everywhere in your body, mitosis happens, except in your reproductive organs where you have meiosis. So that's, see both of them. Forms gametes. Remember, gametes are just your either your egg or your sperm. So those are haploid. So that's what meiosis makes. That's the whole point of meiosis. Involves the separation of homologous pairs. Remember, in meiosis, your homologous pairs separate, and then your sister chromatids separate in meiosis 2. This is meiosis 2. This is meiosis 1. That does not happen in mitosis. In mitosis, your homologous pairs stay together. It's just the copy that separates. So this is B, meiosis. A produces two daughter cells. That's mitosis. They're identical daughter cells. 7, the haploid number of chromosomes in human cells. That's always 23. So you have 23 different chromosomes. One from mom, though, and then the second from dad. When you have one from mom and a second from dad, those 23 different types become 46. That's your diploid number. Okay? So your sperm and eggs have 23 chromosomes, while all of your other cells, those are called somatic cells, have 46. 9. During which phase of the cell cycle is an exact copy of each chromosome made? That's interphase. Remember, interphase is preparation for either mitosis or meiosis. That's when you have one chromosome and a copy of it is made, and that becomes the sister chromatid. So that's always an interphase. That hasn't changed. The process of gametogenesis, so that's the production of gametes, that's a meiotic process. That's made in gametes are made in meiosis. Which cells are formed by meiosis? We already went through this, sperm and eggs, which is nice. Cells made during meiosis have blank the number of chromosomes. Remember, in the beginning they're diploid. Okay, at the very end you have 4 haploid cells, which means you have half as many, because you go from 2N to N, half as many chromosomes. Okay. A cell has 4 chromosomes. After mitosis, each new cell will have, it still has 4 chromosomes. The whole point of mitosis, you go from one cell to two daughter cells that are identical to the parent cell. So if the parent cell had 4 chromosomes, well, your daughter cells still have 4 chromosomes, but you now have 2 cells that have 4 chromosomes. All right, the next, the result of mitosis is 2 new cells, and remember, they're identical. The result of meiosis is 4 new cells. They go from diploid to haploid, so they're not identical at all. And that's the whole point of genetic diversity, that you have 4 different options that can then go and combine. So your eggs and your sperm can go and combine in any fashion that they want to make a zygote, which is the baby. All right, 16. The point of attachment between 2 strands, that is called your centromere. That should be review. And just to scroll down, that's your centromere right there, and that's a centromere right there. 17. What process is shown below? That's crossing over. So remember, when these guys hold hands before crossing over happens, that's called synapsis. Okay? Once they actually, and this doesn't show it well, but it should show it on your own paper. Once they actually flip little sections of their chromosome, then that is called crossing over. Right? 18. What 2 processes are represented by A and B? So here I'm going from 4 chromosomes to 2, so from diploid to haploid, which means that is meiosis, and this represents a sperm. This is an egg. When they come together, that process is called fertilization. So it's B. All right, during normal cell division, which event which series of events is chronologically correct? Okay, so if you think about it, the first thing that happens, you have a cell, and no matter what process you have, whether meiosis or mitosis, okay, first the chromosome has to duplicate, so it's going to be replicated. So 3 is the first one, and that's correct. So I can get rid of B and D. Now, the next thing that happens is that those chromosomes are going to line up in order for them to separate. Okay? And so that's 2. The next thing that happens, they split apart. They go to opposite ends, so that's anaphase. And then here, the nuclear membrane forms around the new set of chromosomes. That is in telophase. So A is your correct answer. 20. If a pea plant's alleles are for height are little t, what's true about its parents? So what I did was I drew this out for you. If it's little t, if the offspring is little t, there are 3 different options that the parents could be. One parent could be heterozygous, and the other one also heterozygous, and right here you have a 25% chance of being little t. Or both parents could be little t, and there would be a 100% chance. For here, one parent could be heterozygous, one could be recessive homozygous, and then there would be a 50% chance. So let's look at these options. Okay, both parents were tall. Well, here that is true. Okay, that's fine, but not necessarily. Both parents could have been short, or one of the parents could have been short. So A doesn't work. Both parents were short. Well, no, these 2 parents are tall. They're heterozygous, so they could create a short offspring. So that's wrong. Both parents contributed a recessive allele. That's true here. That's true here, and that's true here. So that one's okay. Let's check the last one. Both parents contribute a dominant allele. We don't need that. If you look here, you can still have a child that's little t, even though there is no dominant allele in the picture at all. Right, 21. In the Punnett square, which of the following are true of the offspring? All right, if you look here, all of them are going to be tall. Big T, big T, big T, little t. Remember, T is dominant. Tall is dominant. Okay. 22. A male and female bison that are both heterozygous produce an albino offspring. Okay, so you got 2 parents that are big A, little a. Notice they have normal skin pigmentation. So what that tells you is that dominance is in effect. If you have at least one big A, your offspring are going to be normal. And so there's Mendel's rule of dominance. And if you're even thinking for a second, whenever you think, okay, I need to separate my alleles and create a Punnett square, you are using the law of segregation automatically whenever you start to create a Punnett square. So dominance and segregation are working here. 23. When one allele is not completely dominant over the other, this is an example of, for here, it's incomplete dominance. Multiple alleles just means rather than having a big T, little t, you could also have, I don't know, a middle-sized t, for example. But remember, you will always just have 2 alleles. One individual can only have a maximum of 2 different alleles. Incomplete dominance is that mixing of color, so red and white makes pink. Polygenic inheritance tells you that multiple alleles affect just one trait. And then multiple genes, honestly, I don't even know what that is. We haven't talked about that. I mean, it's just multiple genes, but that's not something that we've talked about at all. 24. If you have a black and white chicken which has a speckled offspring, remember that's codominance. Incomplete dominance is the mixing, so that would be a gray chicken. This is actually both alleles, so black and white, are dominant together, so they're both being shown. So they're like co-captains, they're shown at the same time, but it's codominance. 25. Chromosomes come together in a process called synapsis. This is in prophase 1. That's when you also have crossing over. 26. Which of the following genotypes indicates homozygous genotypes? This shouldn't be too bad. Homo means the same, so big A, or little a, little a. Which of the following genotypes show a dominant phenotype? D. If it's big A, or big A, that's what you see is dominant. Which of the following genotypes shows recessive? That one, A. Various forms of a gene are called, these are alleles. Okay, this should be a nice and easy review. If big R is dominant to little r, the offspring of a big R, little r, I wrote it here. Genotype-wise, they'd be big R, little r. Okay, so if you look here, have the same genotype, they don't have a big R genotype, or little r genotype, so those are crossed out. Phenotype, remember, phenotype is the physical characteristic, so what you actually see. And so they would have, a big R, little r would have the same phenotype as big R, so that's your answer. The passing of traits is called heredity. Scientist, sorry, it's blurry, is Gregor Mendel. A phenotype is the physical traits, so this shouldn't be too bad. Okay. A heterozygous father and a homozygous recessive mother. So the father is big F, little f. Make sure you just read the question carefully, guys. It really shouldn't be too bad. Mother, since she's homozygous, recessive, it would be little f, little f. The daughter, if she had no freckles, because freckles is dominant, she would have to be little f as well. Okay, I'm going to do a cross now. So remember, I'm using the law of segregation. Big F, little f for the dad, little f for the mom. Just be careful that you can tell the difference between your F's. Okay, so 50% will be heterozygous, and there's a 50% chance that it will be homozygous recessive. So that's A. If the parent had 12 kids, how many should they expect to have freckles? Well, that's 50%. So 50% of 12 is 6. 39. The type of inheritance, so red and white together to make pink, is incomplete dominance. Okay, so these two chromosomes are homologous chromosomes. Remember, this one's from mom and this one's from dad. Okay, so although they have the same marks for where they have genes, those genes might be slightly different. So one might be big T, the other might be little t. Okay, so the same location for the gene, same type of gene, but different, but being different, right? So either for height, they're both the same gene, but one's tall, one's short. That's called an allele, just to remind you of all these terms. The two lines, so these two are called sister chromatid. The farther apart two genes, so this is the hardest problem, the hardest multiple choice. So keep in mind, the farther apart, if I scroll back up here, the farther apart two genes are, okay, the more, if they're on the same chromosome, if they're linked genes, the farther apart they are, the more likely one of them is going to be crossed over to the other chromosome. So if one gets crossed over, that means that the two genes are no longer linked, that they won't be inherited together. So if you read this question, the farther apart two genes are, the more likely they're going to be crossed over, which I just said, which means the less likely they're going to be inherited together. So that's A. And notice I starred this. This is one of the hardest questions. So go back, re-listen to my explanation. If you don't understand it, come in and we can talk about it. Alright, write the genotype for a purple flower, short pea plant, and a white flower, heterozygous tall plant. So I've got a purple, short, right, and a white, tall. Now, for my crossing, let's say for dad, I'm going to do the one up top first. Big P could go with little t, this big P could go with that little p with that little t, or that little p with that little t. So that's what I wrote right there. For this one, little p with big T, which I wrote there, little p with little t, which I wrote right there, little p with big T, which I wrote there, and little p with uh-oh, I messed this up. Hold on. Yep, I messed this up, guys. I'm sorry. So this should be a little, sorry, I'm gonna think on the ball of my feet. This should be a little p, little t. So this should be a little t, this should be a little t, that should be a little t, and that should be a little t. I'm sorry, guys, because that little p goes with that little t. I didn't think. So what you do then is you combine them. So big P with little p, big T with little t, which I did right there. And that's what I did for the whole thing. If you guys need help with this, except for this bottom row, which is messed up because these should all be little t's, you need to come in. Okay? And then the only thing that's going to change is the number, so that don't look identical. So if it's purple and short, it's not identical. The parent, so these two would be identical. So we can get rid of those two. And I think it ends up being, instead of 8, which I said, I think it ends up being 6. Unless I made a mistake, which I don't think I did after that point. So this should be 6. How many of them do not look identical to the parent? Meaning, how many of them are not purple and short or white and tall? And so you can count that out. Okay. Mendel used the pea plant. This was his model system, guys. Remember, mice, guinea pigs, anything like that are easy to take care of, but that show the traits that you want. Alright, for this, all of this should be really easy, I'm hoping. Prophase 1, they're homologous chromosomes. Metaphase, they line up. The homologous chromosomes line up. Anaphase, the homologous chromosomes separate. D was funky because it includes a lot of different possible steps. It's telophase 1, it can be cytokinesis, and it can be prophase 2, getting ready for the sister chromatid now to line up. And then in anaphase 2, that's when the sister chromatids separate. Okay, so make sure, go back, look over the notes for meiosis to make sure you understand that, because I might ask a question or two about that. And I think that's it, guys. Email me if you have questions.